Okay. Good morning and good afternoon, everyone. I'm Cherry Kubota. I'm a professor at The Ohio State University. I'm the host of this series of webinars, monthly webinars. Welcome to Indoor Ag Science Café, November. This seminar or webinar series is supported by three project groups. One is original one, OptimIA team, led by Michigan State University. The new additional teams, one advanced CEA led by Rutgers University and Ohio State, and CEA HERB led by also Michigan State. Those three organizations, or three project teams are organizing this monthly webinar series to provide scientific technological information to advance uh, controlled environment crop production. Okay, so that's good. Welcome everyone again. I appreciate connecting this. So I like to advertise a little bit before introducing today's speaker. Again, we're gonna record the speaker presentation, but we stop recording afterwards. So feel free to ask questions during that time. But anyway, so the one advertisement I have, Advanced CEA team is offering another, well, it's a similar course, but updated information. It's a course about introduction to greenhouse environment control for crop production. You have opportunity to take this course in a 13 weeks long format starting on January 9th. Two lectures every week. Eastern time, it's a Thursday night. It's recorded, so you can actually take online also. Um, if you are one if you are affiliated with any of the project teams affiliation, specifically Cornell University, Rutgers, Ohio State, University of Arizona, and then a couple of companies also, you have a much reduced registration fee. So just let us know if you are affiliated in one of those project related members institutions and then interested in learning this. This is a great opportunity to better understand greenhouse climate control and sensors and variables and control logics. So it is very hard to find this kind of course, actually, even in university setting. Not many universities are offering this kind of course. So I think it's a really good for professionals, students who are interested in strengthening the understanding of that. So anyway, sorry, it took a little longer than I thought. But anyway, so that's the advertisement. We have three scheduled Indoor Ag Science Cafe webinars coming up. Today is Dr. Roberto Lopez from Michigan State University, who is going to introduce the CEA I should have space in between CEA project. And then December 3rd, Advanced CEA is hosting wavelength selective covering and PV integrated photovoltaic integrated greenhouse systems. And then speaker is Dr. Mura Kachira, University of Arizona. And then January 7th, another Advanced CEA hosted seminar or webinar regarding state of industry by Advanced CEA. So this is very unique topic. And then I think it's an interesting one. Subtitle, Truth Behind the Data. So Dr. Gil Signorini, economist from Ohio State University, and Chris Higgins, America's co-present this topic. We are gonna start 1 hour earlier than usual. This is still Tuesday, but 1 hour earlier than usual because of the speaker's conflicting schedule. So with that, I'd like to introduce Dr. Roberto Lopez. Dr. Lopez or Roberto is also a member of OptimIA, so has been contributing to the indoor agriculture project significantly and then generated quite interesting outcomes. And then now leading separate project, which is more focusing on herbs. So I'm so glad have this opportunity with Roberto talking about the new project. And then so thank you for having this opportunity, Roberto. I really appreciate. So I'm gonna
Thank you.
stop my screen
All
and have
right.
you share.
Well, thank you very much, Cherry. Thank you for the invitation, and it's my pleasure today to talk to you about a relatively new USDA SCRI project that was funded in late 2022, at the same time that Cherry's advanced CEA was funded. So it's great to see that the USDA is committed to the controlled environment industry. I'm going to give you just a brief background because with a lot of these projects, it takes a while to really get them funded. And this goes back all the way to 2017 when Eric Runkle and I were brainstorming on a proposal based on culinary herbs. So half off Michigan beer was definitely helping us out with that brainstorm. So that year, or excuse me, the following year, in 2018, we applied for a planning grant through the SCRI project, and we actually had a meeting in Dallas, Texas, where we were at the State Fair of Texas. Chris Higgins helped us out with that. The following year, based on the input that we got from the industry, we launched a national needs assessment survey. And so that's really what gave us the groundwork to prepare a USDA SREP project. So that was funded again in 2022. And so our team consists of 6 different universities as well as the USDA. So we have 4 individuals here at Michigan State, 2 at the University of Tennessee. Then we have Brian Whipker at NC State, Chris Curry at Iowa State, and at Texas Tech, we have Angela Walla and then Jennifer Bolt at the USDA ARS. I'd also like to thank our advisory board. Our advisory board is made up of quite a diverse group of individuals and companies, some of which you may know, and they definitely help us steer us in the right direction in terms of our project. So again, thank you to them. Okay, so our project has 3 objectives. The first one is really focused on marketing and economics, where we're trying to determine what is the demand for culinary herbs and how can we help the industry, right? By determining what consumers are looking for in terms of the origin, in terms of the flavor, aroma, and how to differentiate these products, as well as looking at the packaging. In objective 2, which is our largest objective, we are looking at production inputs when it comes to environmental control. We're also looking at disease management and post-harvest physiology, because as I'll show you later on in the presentation, there are several culinary herbs that definitely tend to have issues in terms of post-harvest shelf life, especially if they're exposed to temperatures below 50 degrees Fahrenheit. And then probably our most important one that we often tend to forget, especially when we have a transition from, let's say, a floriculture industry to a greenhouse that's producing culinary herbs, and that's food safety. Then our third objective is really that outreach component where we're going to develop different resources for greenhouse growers in terms of how to produce high quality culinary herbs. That's either potted or fresh cut, how to make them more flavorful and nutritious, extending the shelf life, and of course, determining what the consumers really want. So again, our overall goal here is to increase both local and year-round controlled environment production of culinary herbs to meet the domestic demand. Because currently, the domestic demand is not being met. So we're having to import quite a few culinary herbs from throughout the world. And that, of course, reduces the overall shelf life. So what I'm going to do today is basically go through the various objectives and give you just a really two or three slide tidbit of each of the various projects that are being conducted by our team. So as I said before, objective one really focuses on marketing. And so with this component, our group is wanting to really look at the demand and acceptance of both potted and fresh cut culinary herbs, and looking at how there's different value added attributes, right? So is the consumer really looking for a locally grown, grown in the US? Are they looking for enhanced flavor for particular colors, etc.? Then the next component is to look at sensory characteristics that identify both the key taste and scent profiles, so that then we can tell the industry, that in terms of, let's say, basil, consumers are looking for basil that maybe doesn't have such a strong licorice flavor, but they're looking for one that's maybe a little more moderate. And lastly, we're going to identify cues and product information used by different consumers when they're making their purchasing decisions. So this is work that's being led by Bridget Behe and Elisa Ryan, where they're using eye tracking technology to really determine what the consumer focuses on when they're either in the grocery store or in a big box store, or when they're purchasing herbs. Okay, so we are going to look at what Alicia, Carlos, and Bridget have been working on over the last year. And really their overall goal in the research that they've conducted over the last year is to determine, again, those product attributes, the consumer segments that are purchasing culinary herbs, what they're looking for, and most importantly, what they're willing to pay. So they conducted a very large survey across the US, nearly 2,500 participants. And then they gathered the information, again, of their behavior in terms of how many or what their purchases are, what their preferences are, the demographics, and of course their willingness to pay. So here we are looking at, again, factors influencing monthly fresh herb expenditures. And let's focus here on both the winter and the summer months. We can see here that we have consumers that really don't spend a whole lot on culinary herbs. Then we have the medium category and those that spend over $10.50. So what we can see is that we can have about 5.05% increase. And we see this actually when consumers are cooking meals at home, right? They're going to obviously purchase more culinary herbs. And where do the vast majority of consumers, especially those that are in the high category, where do they purchase most of their herbs? And interestingly, direct farms, so this would be considered farmers markets, etc., as well as garden centers. And surprisingly, the lowest percentage were really in the grocery store. Okay, another interesting thing that they have been looking at is to determine if consumers that purchase meal kits, what their tendencies are to purchase culinary herbs. So what we're looking at here is, again, different variables such as food risk. Are these consumers curious? Are they going to expand their palate and cook ethnic foods? Or are they more curious about foods? How much do they spend on culinary herbs? So we can see that individuals that subscribe to meal kits are those that are going to spend more on culinary herbs. But of course, they are a very small and niche proportion of the overall market. As we can see here, of those 2,500 participants, only about 187 subscribed to meal kits, and the vast majority of the other were non-subscribers, and they spent on average about $9 per month on culinary herbs. So again, Alicia and Bridget are focusing on a wide variety of factors, which we will share in the near future. In objective 2, again, we're looking at production, how to increase quality, shelf life, and minimize disease, and obviously increase food safety. And you're probably wondering, why is our team not focusing on insects? Well, when we did our needs assessment, it was actually surprising to us that insects were actually ranked quite low in terms of research needs. So in this objective, we have various tasks. And so what I'm going to focus on in this section is looking at how the air, as well as the nutrient solution temperature, and in some instances, carbon dioxide, how they interact to influence growth, development, and post-harvest performance of fresh cut herbs. We're then going to look at how UV light impacts disease incidence, but more importantly, stem elongation. With a lot of these potted herbs, there are no labeled growth regulators. And so what often happens is these culinary herbs can get too tall. They're not going to fit on a shipping cart or in a box. And then the grower has to dispose of them. And so, again, we're looking to see if we can use a non-chemical way of controlling height. And then lastly, to develop photosynthetic response curves so that we can determine what are the limiting factors or what should we focus on. Should it be increasing the temperature, increasing light, or carbon dioxide? So let's first look at some of the work being done here at Michigan State by Bridget Knight and Eric Runkle. They're focusing on young plant production of culinary herbs with a focus on indoor production, and then comparing that to culinary herbs, young plants that are produced in a greenhouse. So again, they're looking at primarily lighting to determine how they can promote rapid growth, but again, compact growth. So here's some preliminary work where we're looking at cilantro transplants. Again, these were grown indoors under various light qualities. So they provided 210 micromoles of light over a 16-hour photo period. And then these numbers represent, again, that 210. So of course, here they use 20 micromoles of blue light and 190 micromoles of red light. Obviously, we can see some quite stark differences. When we only use blue and red light, we have very compact seedlings, even if we add 60 micromoles of far red light. But here, interestingly, when we reduce the amount of red light and we increase the amount of blue light, we actually see quite a bit of stem elongation. And of course, in these scenarios, it would be very dependent, right, on whether you were growing a potted herb or a fresh cut herb in terms of whether you want that stem elongation. Okay, the next project that we're going to quickly talk about is looking at the interaction of both air and nutrient solution temperature on the production of culinary herbs. And this work is being conducted by Spencer Givens and Kelly Walters at the University of Tennessee. So again, what they're trying to determine is, how do these two environmental parameters, air and nutrient solution temperature, impact the growth and development of these herbs? But more importantly, how do they affect the mineral nutrition and the production of volatile compounds? So thus giving herbs their unique flavor as well as the aroma. So here we have a slightly complicated figure where we're looking at the nutrient solution temperature as well as the air temperature. And obviously the most important thing is, right, that harvestable fresh mass. So let's start off with a relatively cool air temperature and a relatively cool nutrient solution temperature of around 59 degrees. We can see that our harvestable fresh mass is quite low. We get about 6 grams. Then, if we increase the nutrient solution temperature to around, let's say, about 73 degrees Fahrenheit, we see that we gain an additional 7 grams of biomass. However, if we continue increasing the nutrient solution temperature, we actually see a reduction in fresh biomass. Now, of course, this is when the air temperature is relatively cool, around 68 degrees. Now let's look at what happens when we increase that root zone temperature, or excuse me, that air temperature to around, let's say, 86 degrees Fahrenheit. We see that we have a very large increase in the fresh mass. However, if we reduce the root zone temperature, we actually see a decrease in fresh mass. And if we continue to increase that nutrient solution temperature at a high air temperature, we see very negligible increases in fresh mass. So again, Kelly and Spencer are going to be looking at more culinary herbs. And here at Michigan State, we're going to increase yet another factor, which will be carbon dioxide concentration. The next study that I'm going to talk about is developing photosynthetic and carbon dioxide response curves for culinary herbs. And this work is being conducted by Jennifer Bolt and Forrest Brown at the USDA ARS. So again, they're going to look at how plants respond to increasing light intensities, temperature, and CO2 concentrations to really tell growers what they should invest in for particular herbs. And then they're going to model these responses and look at how, let's say, increasing CO2, how much that's going to cost, and really what are you going to gain in terms of harvestable fresh mass. They're also going to assist the project team with analyzing leaf tissue for nutrient analysis. So here's some of their preliminary work where we're looking at 5 different culinary herbs. And probably one of the most important things that I want you to take from this figure is that the CO2 compensation point for most of these herbs was around 22 to 30 micromoles per mole. And what this means is that at that concentration of CO2, the plants were just beginning to photosynthesize. Anything above that, and obviously they were going to photosynthesize. So at ambient CO2 concentrations, which are slightly above 415 micromoles per mole, they yielded a net photosynthesis of anywhere from 51 to 61% of the maximum potential photosynthesis. If you were to inject CO2 to 800 micromoles, they found that you could increase that net photosynthesis up to 72 to 82% based on the particular culinary herb. So here in this instance, we are seeing that increasing CO2 is going to increase your yield. Okay, the next study, again, as I mentioned before, with potted culinary herbs, as we can see in this photograph, they can get quite leggy very quickly, especially if, let's say, the weather changes and the growers are not able to ship out their product. So here we're looking at UV light on potted basil, but we're also looking at other herbs. And this work is being done by Seth Benjamin and myself here at MSU. And our objectives here are to determine if UVA light can be used on a wide variety of culinary herbs, and to really determine at what stage of development is that UV light going to affect that stem elongation, as well as determining what the most effective duration of UV exposure is. So again, here are some of our results. We do have a lot of treatments, but really what I'd like you to focus on is this pot that received UVA light for 4 weeks, where this plant was grown under long day conditions in the greenhouse. So we can see that we definitely can reduce that stem elongation by providing 4 weeks of UV light. But again, we wanted to determine, can we reduce the amount of UV exposure? So here in these 2 examples, we can see that plants received either 2.5 or 3 weeks of UV radiation early on, and then followed by being placed in a traditional greenhouse environment. And again, we found that plants were very compact. Interestingly, with basil nufar, we are finding that it is not as affected by UV light as Genovese. So again, what we're going to look at in the future is look at other wavelengths as well as even look at UVB. Okay, we are now going to change gears and look at Objective 2, Task 2.2. We're here, this objective is really going to, or excuse me, I should say sub-objective is going to focus on nutrition. So we're going to look at conventional, both water-soluble as well as controlled release and organic fertilizers and how they affect post-harvest growth in container grown herbs. We're also going to look at the appropriate nutrient solution pH ranges for hydroponically grown culinary herbs, looking at silicon and calcium to see how they might affect post-harvest shelf life. And then lastly, probably one of the really important things for the industry is to determine critical leaf tissue nutrient ranges, as well as what the various micronutrient deficiencies, as well as toxicities and what they look like for herbs. So again, in terms of the nutrient deficiency disorders, this work is being done at North Carolina State by Danielle Clade and Brian Whipker. And their goal here is to produce not the most attractive culinary herbs, but really they want to induce and record the symptomology of various nutrient disorders. Here are the nutrients that they're going to be looking at on 6 different herb species. So this is their system where they either take out a particular element or they increase it to maybe induce a toxicity. They're then going to develop a photographic guide to aid growers in the diagnosis of various nutrient disorders. They're also going to develop standards for the critical leaf tissue concentration of these various elements. So here's just a snapshot of some of the work that they have started. We're looking at cilantro. And similar to lettuce, for example, we can see that cilantro can develop tip burn when there is a calcium deficiency. You can see with some of the other culinary herbs where maybe you don't see the traditional symptoms of phosphorus deficiency in terms of purpling, but you definitely see stunting of the crop. Okay, the next study is quantifying the effects of pH in hydroponic systems. So this work is being conducted at Iowa State by Hannah Kramer and Christopher Curry. And here, again, they are trying to determine suitable nutrient solution pH levels for culinary herbs. A lot of the current recommendations have been based on other leafy greens such as lettuce. And so again, here they're wanting to determine where that pH should be for various herbs so that we don't have micronutrient deficiencies as we can see here. So here we're looking at basil, dill, and parsley, and looking at the crop responses to a wide range of pHs, anywhere from 4.5 all the way up to 7. And as we can see here with parsley, as that pH increases above a 6, we actually start to see that our fresh mass decreases significantly. So again, this work is going to be quite useful for herb growers that produce fresh cut product. The next part of Objective 2 is Task 2.3, and this is really to develop best management practices to limit diseases on culinary herbs. So this work is being conducted here at Michigan State by Madeline Anthony, as well as Mary Hosbeck. And what their goal is here is to, again, determine what culinary, or excuse me, what pathogens are affecting culinary herbs, and even some that have not been identified, Also working with IR4 to expand the fungicides that can be used on culinary herbs, because currently there are not very many that are labeled. And so what they are doing is they are collecting plants from various retail outlets and looking for various pathogens. So thus far, they have quantified 18 different isolates from 15 different plants, and they're looking at identifying different morphological characteristics for different diseases. So for example, they have found that mint can be affected by Xanthomonas, and these are the symptoms that that mint has when it has Xanthomonas. Again, in some instances, they're actually finding pathogens that had never been reported on these herbs. So again, this is obviously something that you do not want to have on your crop, but this group is going to develop, again, a pictorial guide to the various diseases. And lastly, we are going to look at task 2.4, where this task is going to focus on post-harvest issues. So here, Kelly look at end of production lighting or greenhouse temperatures and how that affects the post-harvest life of herbs.
Sure.
They're going to look at calcium and silicon and how it again influences post-harvest performance, looking at different nitrogen forms, as well as how to increase the shelf life of culinary herbs in retail settings. So the first study, again, is being done at Iowa State, and this is looking at the influence of fertilizer type and concentration on containerized herbs. So this is looking at during production, during retail, as well as in the consumer home. So really, they're wanting to look at, some consumers want an organic product, while others are not necessarily concerned by that. They're more concerned in how many times they can harvest from a potted herb. They also want to determine the best source and concentration of fertilizers and how that's going to extend to the consumer. So they're looking again at both conventional and organic sources in terms of water soluble and controlled release. And they're looking at different rates with water soluble from 100 to 300 parts per million, and with controlled release anywhere from 0.25 all the way up to 0.75 kilograms per meter cubed of nitrogen. So here are the different fertilizers that they are working with. And again, here are some of their preliminary results, where we can see that again, the conventional, whether it's a water soluble or controlled release, look quite well, with maybe slightly more compact with the CRFs. And with the organic, we can see not so much of a difference between the water soluble and the CRF products. This is how the plants look during retail, so we can start to see some stretch across the board. But here are the results in terms of the consumer environment and how those fertilizers, again, performed. Okay, the next part is looking at silicon and calcium to help with abiotic stresses. So this work, again, is being conducted by Jennifer Bolt and Forrest Brown at the USDA. So they want to determine if silicon and calcium can be used to reduce stresses such as chilling injury, low temperatures, especially in basil. And they want to look at this in both potted as well as fresh cut herbs. So here they are looking at, again, the application of silicon to potted herbs. And we can see that the herbs were exposed to 4 and 8 degrees Celsius. So again, about 40 to 46 degrees Fahrenheit. And we can see that when silicon was added to this potted plant, the severity of chilling injury was significantly reduced. So again, we saw that at 4 degrees, we saw a very large impact on the photosynthetic efficiency. So here we're looking at chlorophyll fluorescence. So when silica was added, we see an increase in that chlorophyll fluorescence, indicating that there is less stress. At a warmer temperature, again, we see that that silicon again reduce the chill and injury symptoms. Now here we're looking at again the application of silicon to hydroponically grown fresh cut herbs. Again, these were grown in deep water culture for 3 weeks and they either received silicon or they did not. Then they were harvested, placed in clamshells, and again stored at 8 or 4 degrees Celsius. And they are currently evaluating the weight loss, symptoms of chilling injury, incidence of botrytis, any sort of yellowing, and the overall appearance of the basil after 35 days. And our last objective is that, last but not least, and really a very important component of production, is looking at food safety. So what this part of the team is looking at is really developing protocols for both greenhouse fresh cut and potted herbs. They're looking at developing a needs assessment survey and really developing food safety guidelines for growers across the board, whether they're small, medium, or large, and also looking at geographic regions of the U.S. to develop those protocols. So again, this work is being done at Texas Tech University by Grace Akumo, as well as Angela Walla. And they have quite a few objectives. Again, they want to develop a food safety tool specific to culinary herbs, where then they're going to develop supportive guidance on how to document their food safety protocols. They're going to, again, visit culinary herb producers in several parts of the U.S. in this coming spring. And then they're going to determine intervention strategies to prevent infection of pathogens that not necessarily affect the herbs, but that affect humans. And so here is some of their initial work looking at the survival of Salmonella that has been introduced into either a deep water culture or NFT hydroponic system. Again, this can easily happen where, again, it could be introduced by animals or even by humans. And so what we're looking at is the Salmonella counts on the roots. And so again, these were inoculated. And at day 1, we can see that obviously we have high counts of Salmonella. But by days, let's say 3 to 12, we can see that, especially with the deep water culture, that the roots still have that Salmonella counts. And again, even up to 4 weeks later, where again, whether it's the deep water culture or the NFT. Now, what about that nutrient solution, right? If the Salmonella is on the roots, then they're more than likely going to be on the leaves. And what about the nutrient solution again? So we can see that by day 3 to 12, we definitely see, again, that in deep water culture, we have higher counts compared to NFT. And by 28 days, again, we still have that Salmonella in the nutrient solution. So again, this is going to help Angela develop protocols on how growers can mitigate the introduction of these pathogens that can really cause a lot of illness and sometimes death and very costly to the herb industry. Our last objective, again, is focused on outreach. So we're going to develop different things to really get that information out to growers. So we're going to develop print and electronic publications, webinars, podcasts, and videos. We're also going to do something unique where we're going to host online office hours where growers can basically ask questions or submit pictures of samples, let's say a particular nutrient disorder that they're seeing or a pathogen. We're going to develop lots of different in-person presentations, workshops, and grower trials, and really try to engage with our stakeholders and disseminate the results in a way that they're applicable. So we have developed a website. Again, this website is still in development, but there is currently information on there. And the URL is scri-ceaherb.org. As Sherry mentioned earlier, we are, again, working with the OptimIA and ADVANCEA HERB to increase the content in the Indoor Ag Science Cafe to really look at not only leafy greens such as lettuce, but really to introduce culinary herbs into the mix, as well as Cherry's component in terms of the engineering of indoor and greenhouse production. One thing that I really wanted to share is that we are starting a article series. So if you want to learn more about a lot of the projects that I very briefly talked about today, this part of the series is going to be in Greenhouse Management Magazine, where again, we're going to focus on a lot of the different topics that I've talked about thus far. Another series is going to be placed in Produce Grower Magazine, and this is going to be a much larger one. For example, where Brian Whipker is going to cover the various nutrient disorders of basil, mint, and cilantro. And again, some of the other marketing and food safety projects. And I know I went pretty fast. There was a lot to cover. But again, if you have any questions, I'd be more than happy to answer those at this time. And one thing that I forgot to mention is that various team members are going to also participate in the Indoor Ag Science Cafe, where they'll present more of their research in depth. So that will be coming in 2025.
Great. Well, thank you so much, Roberto. A lot of information, a lot of projects, very exciting. I really appreciate sharing that various outcomes so far. So excellent.